Welcome back, everyone. So we're going to continue on with Chapter 9, Biotechnology. So in our second section of biotechnology, we're going to take a look at biotechnology in medicine and agriculture. So in 9.2, we're going to take a closer look at basically the applications of biotechnology in medicine and agriculture. So by the end of this video, you should be able to describe the uses of biotechnology in medicine, as well as describe the uses of biotechnology in agriculture. So we're going to take a closer look at what genetic testing is all about and how it's performed to identify disease-causing genes, and how it can be of a benefit for people that where we have certain certain genetic diseases that run in families. They could be tested for these genetic diseases even if they haven't developed symptoms yet, and perhaps get gene therapy to cure those genetic diseases before they even started. So we'll talk about the medicine aspect, as well as the benefits of biotechnology in agriculture, where we can actually use transgenic organisms to produce crops that are more resistant to diseases, or we don't have to use pesticides on them and things like that. So we'll go through all of that biotechnology. So let's start out by talking about their uses. Okay, so biotechnology in medicine, again, it can allow us to give us a genetic basis of heritable diseases, and then we can use the technology to manipulate and fix mutant genes. So we'll give you some examples of that in this video. Also in agriculture, we can enhance resistance to certain diseases in our plants. It can resist certain pests, like different insects, and even environmental stresses. And all of this will then improve crop yield, so that we can enhance the quality of our crops as well as the amount of the crop, and that will allow us to produce more food for our growing population in the same amount of space. Okay, so there's definitely good uses for this biotechnology, not only for the field of medicine, but the field of agriculture. So we're going to start out with the field of medicine first. So let's talk about genetic diagnosis and gene therapy. So first off, let's take a look at genetic testing. So genetic testing involves identifying gene variants in an individual that may lead to a genetic disease in that individual. So some of you may have heard of genetic testing before. So you may have heard of, for example, like the BRCA gene. So the BRCA gene is, or there's actually a couple of them, the BRCA genes. They are a couple of genes that if you possess those genes, they increase your likelihood of developing certain cancers, particularly for females, cancers of the breast and ovaries. For males, they can also increase the likelihood of cancers of the breast, but also other types of cancers. And so if you are positive for those genes, if you possess those genes, then knowing that you have those genes, you can take preventative measures. So this could be, for example, perhaps gene therapy. So gene therapy is a relatively new form of medicine. So it's a technique used to cure heritable diseases by replacing mutant genes with good genes. Okay, so you can see in this diagram, this is directly out of your textbook. So what we do is we introduce the non-mutated gene into a virus. So in this case, it's basically an adenovirus so we introduce that, we remove the DNA or RNA from the virus, and we replace it with a non-mutated gene into the virus. And viruses, what are their jobs? Basically, their jobs is to attack and bind to a cell and insert DNA into the cell, right? And trick that cell into making that DNA. And so they're perfect for this type of therapy. So they're injecting that non-mutated gene into the cell. So in this case, what they're doing is they're injecting the cells that are actually diseased. So in this case, the virus, the vector, is introducing the disease cells with the non-mutated gene, and so we're delivering the gene that actually produces the correct protein or the correct gene into the genome. Okay, and so this is going to kind of correct the mistake. And so this is really new. So gene therapies, they're primarily in the experimental stages. Some have been successful, but it's still in the primary stages. So it's not like it's readily available for everybody. But probably in the next 20 years, hopefully this will be something that is more and more often done for people. Okay, so that's something that you can look forward to seeing more and more common, especially if you being probably younger, and if you do have a history of breast cancer in your family, and that perhaps your family does possess the BRCA gene, perhaps there is gene therapy that is something that will be available to you within your lifetime. Okay, so that is something that might be available, and as well as other types of gene therapy for other common mutations that are out there. Okay, so that's something that we can use. That's becoming more and more present. We can also use biotechnology for the production of vaccinations, antibiotics, and hormones. So let's talk about vaccinations. So traditional vaccines are just basically weakened or inactive forms of microorganisms or viruses to stimulate the immune system. So basically we take like an inactive form of the rhinovirus, for example, and give it to you, and then that kind of tells your body like, oh, this is what it looks like, and then it kind of stimulates your immune system to produce antibodies against it. So that's the traditional form. Our more modern techniques involve, instead of giving you like an inactive form or a weakened form of the virus, we just give you specific genes of the microorganisms, and then mass produce it. And so it's less likely that people with like compromised immune systems will react to it, or it's less likely you're going to get an inverse reaction. So, and it also allows us to get to produce vaccines that are more broad. Okay, so because how, for example, with the flu is constantly mutating. Okay, so that's why there's like every year we have to kind of predict what strands are going to be showing up in our population, and sometimes the strand that the vaccination that is produced is right on point, and if you get the vaccination, you're protected. But sometimes it's not the prediction is incorrect, and a lot of people end up getting the flu even though they have the flu vaccination. Well, with this new modern technique, the genes get cloned from the virus, and it helps prevent it's more broad protection. And so even if the flu is mutating, because it involves the genes, it covers more broad-based, even the evolving strands you're protected against those. So that might be a little bit better in the vaccinations that you're getting. So it might last a little bit longer, I should say, in protecting the coverage of the vaccine. Okay, so that's traditional vaccines versus modern. More modern ones might be a little bit more broadly covered. Okay. Production of vaccines, antibiotics, and hormones. When we talk about antibiotics, so vaccines, they can protect you against different microorganisms as well as viruses. Antibiotics, life, they can only protect you against bacteria, okay, or living things. They can't protect you against viruses. So if you go to the doctor and ask for antibiotics because you have a really bad cold, hopefully the doctor will not prescribe you antibiotics, because the common cold is a virus. The antibiotics will do nothing to kill the virus because the virus is not alive. It's considered non-living. So antibiotics, they kill bacteria. Okay, so bacteria, actually antibiotics, they are naturally produced by microorganisms. Most of them are produced by fungi. So for example, penicillin is a natural antibiotic produced by a fungus that grows on oranges. So if you actually see, if you take an orange and you leave it out for too long and you see this mold growing on it, that's actually a fungus that's producing penicillin. Okay, so that's where we actually got penicillin from. And so that we can actually use that technology. Basically we can manipulate fungus into producing different types of antibiotics. Okay, so and so that's where we get the majority of our antibiotics from. Okay, and if you think about it, the fungi, they can't really move, so they need to protect themselves from bacteria. So that's they produce antibiotics to protect themselves. So we can just manipulate the fungi to producing lots of antibiotics to protect us as well. And then finally, hormones. So we've already talked about how we can trick bacterial cells into producing human insulin. So this is just kind of a review of that. So we can take a bacterial plasmid and we can basically isolate the human insulin gene, and using restriction enzymes we can cut the plasmid and creating those sticky ends, and the same restriction enzyme can cut the human insulin gene, and then we can use the DNA ligase to basically glue in, remember the ligase is the gluer, glue in the segment of the human gene into the plasmid to form recombinant DNA, which then gets inserted back into the bacterial cell. In this case, it's E. coli is the bacteria. And then that E. coli will go ahead and divide via binary fission to produce identical copies of itself. And from there, the bacteria will start to secrete human insulin, so we can use that to produce insulin. We could also use the human gene that produces human growth hormones, so we can actually do that to treat growth disorders in children. So it's not, there's other genes that we can use aside from the insulin gene to treat other hormone disorders. Okay, so there's, other things that we can use. All right, so those are some examples of how we can use the biotechnology for medicine. Okay, so we're going to switch over into well, actually, we'll continue with medicine, but we'll switch over to transgenic animals. So with transgenic animals, we can actually have certain genes cloned and expressed in animals. So we've done this with sheep, we've done this with goats, chickens, and mice. And so when we do that, we make transgenic animals. So we've been able to splice in genes into sheep and goats so that they can express human proteins in their milk. And sometimes this can be beneficial. So for example, we can get a blood anticoagulant protein produced in the milk of a transgenic goat. And so that can be used for human use. So if we want a blood anticoagulant, coagulant means that it's going to, coagulate, it's going to bring together. But if we want like a blood thinner, we can do the opposite. So this allows us to, use, be able to produce this blood thinner protein produced in the milk of this transgenic goat, and then we can use that for human use. Okay. This is a picture, like mice, mice are used a lot in research. So these, you can see these are two of the three mice here are transgenic. They have a gene that causes them to kind of glow under UV light. Okay, and you can see, which two are glowing and then which one's not. Okay, I don't really know the reason why they injected it. They probably used, some sort of, I don't know what gene they used, but yeah, so they do a lot of experimentation on mice for, to see what, what genes they will work. But I don't know why this would be of use for humans, but, it was in your textbook, so I used the picture. Okay, so let's switch over to agricultural uses. So this we usually use with transgenic plants. So with transgenic plants, we have a lot of GMOs. So remember, GMOs are the genetically modified plants. And we use that to create desirable traits. And so that could be, for example, disease resistance. So we talked about the rainbow papaya in the last video that is resistant to the ring spot virus in the Hawaiian Islands. We can have GMO plants that have an herbicide in it. Or we can have some that are resistant to pests, like insects. We can have some that have better nutritional values, such as the golden rice, or even a better shelf life that lasts longer. Okay, so those are some desirable traits. So we're going to talk about 3 examples from your textbook of GMO plants. Okay, so the first one is transformation of plants using Agrobacterium tumefaciens. So this particular example isn't really an example to produce like a desirable trait. It's just what we use to get a desirable trait into the plant in the first place. So as we know, plants, they have really thick cell walls. And so it's really hard for researchers to get the desirable trait gene into the plant in the first place. So a lot of our plants, or a lot of the plasmids that we use to get the plant, get the DNA, the desirable DNA into the plant, is from this Agrobacterium tumefacens. So Agrobacterium tumefacens is a bacterium that can cause tumors in plants. So we don't actually want to use, so, it's easy to get it into the plant because it causes a tumor into the plant. So it's used to invading plants in the first place. So we can use that to our advantage. Okay, so we can actually take this particular bacterium that's used to invading plants, and we can trick it into inserting the DNA that we want to insert into the plant. Okay, so this is used to transfer the DNA of a desired trait into the plant. Okay, so we can insert whatever trait that we want. So if we're trying to grow golden rice, for example, we can insert the daffodil gene that enhances the production of beta carotene into the plasmid of the Agrobacterium tumefacens, and then we insert that plasmid back into the bacterium, and then that gets introduced into the plant, and then the plant starts to produce the beta carotene and starts, then we get golden rice. Okay, so this is strictly used, so this Agrobacterium tumefacens is strictly used to transfer DNA. Okay, so it becomes, it's just there to incorporate the recombinant DNA into the plant. All right, so it's just a way that we can basically create the transgenic plant in the first place. All right. The next example is the organic insecticide Bacillus, I can't pronounce the last word, otherwise known as BT corn. So Bacillus thuringiensis is a bacterium. So that particular bacterium produces protein crystals that are toxic to many insect species. So basically what we can do is we can splice in that BT, gene from the bacterium into corn, okay, and introduce that genetic characteristic, that gene, into the corn's genome through that whole process of genetic engineering to create BT corn. And so now the corn itself produces their own crystal BT toxin. And this particular toxin is completely safe for the environment. It's not toxic to mammals, including humans, but it is toxic to many insect species. And so when we plant BT corn, you can see what happens. We don't have to use as much pesticide or insecticide onto the crop. Okay, so corn is notorious, for use of pesticides. We have to spray the crops with a lot of pesticides because a lot of insects like to eat corn. So there's this figure here is showing a great correlation with the increased percent hectares of BT corn planted increasing, but also the decreased usage of insecticide that's being sprayed. Okay, so because the corn itself has its own protection, it has its own pesticide or insecticide ingrained inside of it, so it's becoming resistant to the insect species. So the insect species are not eating it. This has been approved by use for organic farmers. So, if you do eat organic corn, it might be BT corn. Okay, so it might be a GMO. There is an issue though with this particular corn is there are some insects that are evolving to become resistant. So evolution continues. Some insect species are evolving be able to eat the BT corn, even though the BT corn, is producing these crystals that are supposedly toxic, but some insects are developing a resistance to the toxin. Okay, so that's BT corn. And then finally we have flavor saver tomatoes. So the flavor saver tomato was the first GM crop introduced into the market in 1994. So this was actually genes, actually there's a couple of genes that were inserted that basically prevented the aging process. So it prevented the softening and rotting caused by fungal infections. At first, when they were first produced, they looked great, but they tasted awful. So some additional genetic modifications were used to improve the flavor. You don't see them on the market anymore because they didn't really ship very well. So they weren't, you don't really see them anymore on the market because of that. They don't ship. But they were the first GMO crop, so that's why we're mentioning them. Okay, so that's the flavor saver tomato. And you can see this one, it says genetically modified seeds here. All right, so we're going to do a review question. So what is the role of Agrobacterium tumefacens in the production of transgenic plants? Is it A, genes from Agrobacterium tumefacens are inserted into plant DNA to give the plant different traits? Or is it B, transgenic plants have been given resistance to the pest Agrobacterium tumefacens? Or is it C, Agrobacterium tumefacens is used as a vector to move genes into plant cells? Or is it D, plant genes are incorporated into the genome of Agrobacterium tumefacens? So what is the role of that bacterium? If you said C, I would agree with you. So Agrobacterium tumefacens is the vector, okay? So it's the plasmid. It, we insert the DNA or the gene into the plasmid, and it's used as the vector to get it into the plant cell. So C is the correct answer. All right, so we are done with section 9.2 Biotechnology in Medicine and Agriculture. So by now you should be able to describe the uses of biotechnology in medicine as well as the uses of biotechnology in agriculture. So in our final lesson in Unit 8, our lecture, this is what our 8th video, we'll get into 9.3 genomics and proteomics. I'll see you then.